All right, well, it's noon, so I am going to go ahead and get started. Hi, everyone. Welcome to Grand Rounds. Today is the John Butler Endowed Lecture. We're very grateful to have Dr. Budinger here from Northwestern to present with us. I just want a couple housekeeping things. Send your questions in via the chat through the course of the lecture, and then at the end, I'll read out questions. So ask questions throughout, and we'll save time at the end for them. And then I'm going to step out of the way and let Dr. Glennie and Dior introduce the Butler Lecture Series.
Thanks so much, and good afternoon. Welcome to Medical Grand Rounds and this year's John Butler Endowed Lecture. Before handing the Zoom control to Dr. Rina Mehra to introduce this year's lecturer, I'd like to provide a little bit of background on who John Butler was and why we have a Grand Rounds named after him. John Butler, or JB as everyone knew him, began his medical education at the University of Birmingham in England, where he focused on lung mechanics. He was invited to the Cardiovascular Research Institute in San Francisco, where he worked with Julius Comro. JB was recruited to build the Division of Respiratory Diseases at the University of Washington in 1964. He had a vision for a new subspecialty that included scientific research and clinical excellence, and he deserves a great deal of the credit for the success of our current division. I had the great fortune to have known JB. He was a true English gentleman, very kind, thoughtful, and a creative scientist. We have had a long list of impressive scientists and leaders in our field who have given this JB lecture. Each year, a committee identifies an individual who is an international leader, has a broad interest in pulmonary and critical care medicine, and especially important to have a deep commitment to working with young investigators. I believe this year's John Butler Lecture embodies many of JB's leadership qualities. Over to you, Rina.
Thank you so much, Dr. Glennie. I appreciate that. And what a great pleasure it is and delight to introduce Dr. Scott Budinger as our 2024 John Butler Lecturer. So Dr. Budinger is Chief of the Division of Pulmonary and Critical Care Medicine and Professor of Cell and Molecular Biology at Northwestern University Feinberg School of Medicine and the Ernest S. Bazley Professor of Airway Diseases. He's internationally recognized for his research, where he is focusing on mechanisms by which environmental stresses activate signaling pathways in resident cells of the lung, and also therefore the pathways that induce lung injury and fibrosis. He has a focus on alveolar macrophages and their interactions with alveolar epithelium that then drive forward a susceptibility to lung injury and fibrosis, particularly in older adults. He has extended his research to humans, where he has used multi-omic approaches to further extend this work, and currently leads a team of investigators to focus on how mitochondrial metabolism in particular drives changes in proteostasis, the dynamic process in which cells control the conformation and binding interactions of proteins, and probably has something to do with his chemical engineering background that he has as well. In addition to his research, he has served as deputy editor of the American Journal of Respiratory and Critical Care Medicine, one of the main flagship journals of our field, as well as on the editorial board of the Journal of Clinical Investigation, currently in its 100th anniversary, with a meeting being held at Northwestern. He has a true interest in training the next generation of investigators and physician scientists, and as such has been named as the prestigious Mentor of the Year at Northwestern Feinberg School of Medicine. On a personal note, we shared our journey together at Loyola University Medical Center many moons ago, and we're reminiscing a little bit about that earlier. So it's just really such an honor and delight to have Dr. Budinger here. lecturer, and he will be presenting on why Atlas, how molecular pathology is transforming medicine, and we're very excited to hear from him. Thank you so much for agreeing to do this, Dr. Budinger.
Thank you, Arena and Rob, for that very kind introduction and for the honor of presenting the Butler lecture here at the University of Washington. I really apologize for not being able to be there in person. Seattle's one of my favorite cities in the country, and had we not had the JCI celebration, I would love to be there with you in person. I have no conflicts to disclose. We address in our laboratory this question of why is age the biggest risk factor for the development of chronic and acute lung diseases? And so this was a review article that I wrote with Victor Thanakal, who's chair of medicine at Tulane, and many others some years ago, where we pointed out data that showed that for chronic lung diseases like COPD and IPF, there's an exponential increase in both the incidence and death rates from those diseases with age. And that's also true for the primary acute lung disease, which is pneumonia. And I think this association between age and pneumonia became evident to the entire world during the COVID-19 pandemic. We studied this problem through the prism of a single cell population in the lung, and that is the alveolar macrophage. Alveolar macrophages are a tissue-resident macrophage, and every tissue in your body, every organ, and probably every microenvironment within those tissues has its own population of tissue-resident macrophages. And I like to think of these cells as the house sitters of the tissue. Their function there is to deter invaders. If somebody comes in, they call 911 and bring in the rest of the immune system. They keep things alive that are alive within the lung, and they clean and repair minor injuries that might occur while you're away. And what we've learned over the past decade is that these cells are actually very different from one another depending on their tissue microenvironment. And this is in response to signals that come from that tissue microenvironment that shape the phenotype of these cells. So hopefully, without taking this analogy too far, I've made the alveolar macrophage into the caretaker for the air filtration plant, the microglia for the Amazon web server, the cardiac macrophage for the nuclear power plant, and the kidney macrophage for the water filtration plant. And the simple hypothesis that we test in our laboratory is that dysfunction in these cells, either cell autonomous dysfunction, because some of these cell populations like microglia and alveolar macrophages are long-lived and self-renewing, or the loss of signals from the local microenvironment that shape the function of these cells with advancing age, causes the stochastic accumulation of damage to the tissues. And some of that damage to the tissues is responsible for the phenotypes that we see in the older patients that present to our clinics, hospitals, and our ICUs. So this is, I promise I won't show you too many data from mice during this talk, but this is actually a summary of studies, a lot of studies that we and others have done in mouse models of disease to try to understand the function of these alveolar macrophages and how that changes with aging. So just to give you some ideas about the nomenclature and familiarize you with some of the terms I'll use in the rest of this talk, on the left here, we have these tissue-resident macrophages, these tissue-resident alveolar macrophages. Like I said, these cells are long-lived and self-renewing, and these cells we know are absolutely necessary in the lung for homeostasis. If you lose GM-CSF, the protein that's required for the differentiation and maintenance of these cells in the niche, either because of a genetic mutation or you develop antibodies against GM-CSF later in life, you actually accumulate phospholipids and proteins in your alveolar space and literally drown in your own surfactant, and that disease is called alveolar proteinosis. We also think these cells are important for crosstalk with other immune and epithelial cell populations in the lung, but the evidence for this is actually less clear. During injury, everything changes. So in response to signals from the local microenvironment, including signals that are coming from that alveolar macrophage, classical monocytes are recruited from the circulation into the alveolar space. And within the alveolar space, these classical monocytes rapidly differentiate into what we call monocyte-derived alveolar macrophages. If you look at these cells with something like flow cytometry, they look very similar to the tissue-resident macrophages. But we know from lineage tracing studies that these cells are transcriptionally very, very different from the tissue-resident macrophages in that they express a whole lot more of the inflammatory and fibrotic genes that are involved in lung injury and fibrosis. We also know, importantly, that in mouse models, if we use genetic tricks to delete these cells while leaving the tissue-resident cells intact, we make lung injury and fibrosis generally better, certainly in viral pneumonia and in mouse models of fibrosis. During the injury, these monocyte-derived alveolar macrophages often far outnumber the tissue-resident macrophages. You're going to see some evidence of that later. So during resolution, most of these monocyte-derived alveolar macrophages undergo apoptosis. And the normal niche is restored either by the proliferation of these tissue-resident alveolar macrophages or the persistence of these monocyte-derived macrophages within the alveolar microenvironment. And we have shown that over time, these cells are shaped again by signals from the healing now microenvironment into cells that are transcriptionally and functionally indistinguishable from their tissue-resident counterparts. So in mice, we were able to use lineage tracing studies. We could actually put tags on these monocyte-derived cells and not on these cells to distinguish them. But in humans, you can't do that. And so we were really early adopters of single-cell RNA sequencing, which is one of the molecular pathology technologies that I'll talk about here in this talk. And this was actually our first study, in fact, the first study in the lung that used single-cell RNA sequencing and applied it to explants from patients that were undergoing transplantation. Our prediction from that slide I just showed you was that we would see two transcriptionally distinct or more populations of macrophages in the fibrotic lung. And in fact, that was the case. So what you're looking at here is actually what's called a UMAP. Each dot on this UMAP represents a single cell and is a representation of the several thousand genes that we detect in each one of those cells in two-dimensional space. And what we've asked the computer to do here is just put cells with like transcriptomes with other cells with like transcriptomes. And it's clustered this group of cells here that you see in the middle that have cells from both the donor lung, this is in the aqua, and the fibrotic lung that's in the salmon color in this cluster. But you see down here at the bottom, this cluster of cells that actually is only expressed that is only found in patients with lung fibrosis who had undergone transplantation. And if we look at what genes are in these different areas of the lung, you can see that the maturation genes in alveolar macrophages like MARCO and PPAR gamma and MRC1 are higher expressed in this shared population of tissue-resident macrophages, whereas genes that have been causally associated with fibrosis in mouse models are expressed in this population of macrophages that's only found in the fibrotic lung, largely confirming what we had predicted we would see from mouse models. And so this was a nice example of how we could use single-cell RNA sequencing or molecular pathology in humans to confirm findings in mouse models. But it was as we circulated these data amongst the other researchers in our group, and even with the clinicians in our group that were performing clinical trials using agents that they thought were targeting specific pathways in the lung, that we realized that these technologies were really disrupting science and the way that we do science. And this became more evident in the next year when two other groups published single-cell RNA sequencing data from human lung explants of patients undergoing transplantation. One came from the Vanderbilt group, John Kropsky, and another one came from Naftali Kaminsky's group at Yale. And I just want to point out two things about these data sets. First, they are completely compatible with one another. So you can actually find the exact same things in all three of these different data sites, despite the fact that they were performed at different parts of the country. And the second thing, the second thing is that what I want to highlight is the number of patients here. In total, these studies include only 106 patients, really a small number for a human study. And yet they have had an enormous impact on the field as evidenced by one metric, the number of citations for these studies. And, I think that there's been some resistance to the use of these technologies, but I actually find it quite intuitive that these technologies would provide us with a lot of information. So back when I was training, and maybe when Dr. Glennie was training, we used to learn things from textbooks, and these were two of the textbooks that we, received when we were first-year medical students, and were told that we were going to have a test on it 2 years later. One of those was actually the Robinson-Cochran pathologic basis of disease, where we actually learned pathology, and we learned to link that pathology then to the clinical phenotypes that we observed in our patients, both their history, their physical exam, and their laboratory values. And so really what we're doing here with single-cell RNA sequencing, with spatial transcriptomics, spatial metabolomics, and metabolomics is what we've been doing for centuries as clinicians in medicine, which is doing careful observations of pathology in small numbers of patients, linking that with clinical information, and seeing if we can use that to provide insights into therapies that might benefit our patients. So what I'm going to do for the rest of this talk is really talk about some examples of how molecular pathology is driving discoveries in lung disease. And I'm going to focus in this talk mainly on single-cell RNA sequencing because it's the oldest of these technologies. But I want to point out that our paper in 2019 was the first single-cell RNA sequencing data in the human lung. So this is still a very young field, and yet I think it's already yielded some really important results. The first story I'm going to tell you about is the CSF1 receptor as a target in pulmonary fibrosis. And this is actually one of the first single-cell RNA sequencing studies that we did in our laboratory in a mouse model. We published it back in 2020. And I'm going to just show you the data here. This will be easy for you to understand having seen the human data. So here we're looking again at macrophages on this UMAP. Over on the left corner here, we have interstitial macrophages, but I want to draw your attention to these alveolar macrophages on the right side of the figure. You're going to see the same pattern you saw in the human lung, where you see the salmon and the aqua together in these alveolar macrophages that are shared between the mice that were treated with asbestos particles and mice that were treated with a control particle, titanium dioxide. And then you can see this population of macrophages, that's the little tail here on this UMAP, that's only represented by macrophages from the asbestos-treated animals. And one of the things that we noticed about these macrophages in this population was that they expressed a lot of the CSF1 receptor. And this is unusual because I told you that alveolar macrophages really rely on CSF2 or GM-CSF for their differentiation and maintenance. And you can see the CSF2 receptor is expressed on these mature macrophages that we see in the titanium dioxide treated animals, whereas you have relatively more of the CSF1 receptor in the asbestos treated animals. And so Nikita Joshi and Satoshi Watanabe performed an experiment to see if, like other macrophage populations throughout the monocyte-derived macrophage populations throughout the body, whether these cells relied on CSF1 receptor signaling for their maintenance in the niche. And this made sense to us because the cells that were expressing CSF1, the ligand for that receptor, were cells that were involved in inflammation, like neutrophils, activated T cells, and fibroblasts. And so they treated the mice with either an antibody against CSF1, the ligand, or an antagonist of the receptor, this PLX compound. And they were able to show that they were reducing the number of these lineage-labeled monocyte-derived macrophages. So they decreased those bad guys that I told you about in the earlier slides. And when they did that, they were able to reverse established fibrosis induced by asbestos. Asbestos in mice causes this relatively mild peribronchial fibrosis that persists over the lifespan of the animal. It doesn't get worse, but doesn't get better either. And we were able to show with either of these therapies that this got better. And so we were really pleased to see just a few weeks ago that this paper was published in the New England Journal of Medicine where they gave an anti-CSF1 receptor antibody to patients with refractory chronic graft versus host disease. These were quite sick patients that had failed multiple different therapies. This was a phase 2 study. And they saw a really remarkable response rate. And I would draw your attention over here to the the lung disease, where they saw 16% of patients had a complete reversal of their graft versus host disease in the lung, and 31% had a partial response. This drug has just received FDA approval for the treatment of graft versus host disease, and it's in clinical trials in patients with pulmonary fibrosis. So that's an example of actually how we can use these single-cell RNA sequencing technologies to make the leap from mice to humans in terms of thinking about drug targets. And I want to move to a different example of that and talk some about crack channels as a target for a therapeutic target in patients with SARS-CoV-2 pneumonia. To start this, I have to introduce you to this program. So we were interested in not only looking at these monocyte-derived alveolar macrophages in explants from patients with lung fibrosis, but we really wanted to look at this in our patients in the intensive care unit with pneumonia. And to do that, we established this project that's called the Successful Clinical Response in Pneumonia Therapy, or SCRIPT, Systems Biology Center. This is led by Dr. Rich Wunderink, who was the medical director of our ICU and is now a senior scientist within our group and still leading the SCRIPT project. This project takes advantage of the fact that in our intensive care unit, like I think in your intensive care unit at the University of Washington, when clinicians suspect that a patient might have pneumonia, they have a low threshold for performing a BAL procedure to determine the etiology of the pneumonia. So a bronchoalveolar lavage sample is obtained. That sample goes to our clinical laboratories. We get a multiplex PCR that has 33 pathogens along with antimicrobial resistance genes that comes back within 3 to 4 hours. We get biochemistry and cell counts on the fluid, and within a couple of days, we have quantitative cultures. And we've shown that our physicians use this information to narrow or broaden antibiotic therapy based on the results of this BAL sampling. Some patients in our intensive care unit who are there for longer stays will often have serial BAL samples. And so in some patients, we have more than one sample. In patients that consent, residual biologic fluid that's not needed for that clinical testing comes to the research laboratories, where it's subjected to multicolor flow cytometry in all cases. And then we have a suite of genomic and other technologies that we can apply to the BAL fluid. The main workhorse technologies that we use right now are single-cell RNA sequencing and shotgun metagenomics for the DNA microbiome. We combine these data with data that we're extracting from these patients from the electronic health record, which is quite dense in the ICU. And the goal of this project is to put together models of models of pneumonia pathogenesis that try to explain the observation that 50% of patients with pneumonia fail therapy despite optimized PK/PD antibiotics. And this is just the disposable bronchoscope that we use. And this is a patient who you can see is prone in our ICU, picture taken through the window during the COVID-19 pandemic, who's receiving this procedure. And this work was done by Sasha Misharin and Ben Singer, who are now tenured associate professors in our group. So because the SCRIPT project had started a couple of years before the pandemic, or at least a year before the pandemic, we had already enrolled about 200 patients with known or suspected pneumonia into the SCRIPT project before the first patient with SARS-CoV-2 came to our unit in March of 2020. So we were uniquely in a position to test the null hypothesis that SARS-CoV-2 pneumonia is no different from pneumonia secondary to other pathogens. And this was work that was led by Rogan Grant, who's now a Schmidt Fellow in our group, Luisa Morales Nabrata, who's transitioning from a K to an R, Nick Markov, who's a graduate student in our program, and Melissa Querrey, who's now in our PSTP program. So I'm going to actually give you the punchline first and then go backwards and show you some of the data that informed this model. And so this is the model that we developed based on this integration of the clinical data with the genomic data. And so here I'm going to here's just in panel 1, you have this cartoon of the alveolar space that's relatively simplified. You have this alveolar type 2 cell. These are small cuboidal cells that sit at the corners of the alveolus, and they secrete surfactant proteins and other defense molecules into the alveolar space. You have these type 1 cells, which are very large, flat cells. These lay like a blanket over 2 or 3 alveoli. They're very, very thin. And here I've drawn an outsized tissue-resident alveolar macrophage that we've been talking about throughout this talk. SARS-CoV-2 gains access to the alveolar space through droplet aspiration, where it infects the alveolar type 2 cells that express the ACE2 receptor. And it's important that we know from single-cell RNA sequencing data from the Human Lung Cell Atlas that at most 2% of alveolar type 2 cells express the ACE2 receptor. This becomes important as we talk about this more. This leads to an increase in the concentration of viral particles within the alveolar space, and that infects these tissue-resident alveolar macrophages via mechanisms that we still don't understand. These cells do not express ACE2. These cells then become activated and release T-cell chemokines into the bloodstream. At the same time, we think they're able to activate cross-reactive T cells that are in the alveolar space. And there were about 20% of patients harbored these cross-reactive T cells even before the COVID-19 pandemic. Those activated T cells release interferon gamma, and that causes the macrophages to release even more T cell and monocyte chemokines. Eventually, the interferon gamma induces death of the alveolar macrophages. There was a nice paper that came out during the pandemic that showed how that happened. These cells are replaced by those monocyte-derived alveolar macrophages that we've been talking about throughout this talk. Those cells respond to the interferon gamma and become infected by SARS-CoV-2, causing this feed-forward loop between the activated T cells releasing interferon gamma and the infected monocyte-derived alveolar macrophages that persists until the virus is cleared. So how did we get to this model? And I'm going to show you, first I'm going to go through some of the clinical data and talk about what's clinically different. So here what we're looking at on these panels is actually patients with COVID-19 that are in red, patients with other kinds of viral pneumonia that are in the script cohort, so other viral pathogens, and these are patients with other pneumonia, 99% of them with bacterial pneumonia. On the left here, you have a special group, which are patients that were initially suspected of having pneumonia, but were subsequently adjudicated to have respiratory failure secondary to other causes. If we look at this population, they're all similarly old. They all have a similar male preponderance, and this is known from the literature previously. Our SARS-CoV-2 pneumonia patients were more likely to be Hispanic or Latino compared to the population that is usually in our intensive care unit, and they had a higher BMI. They had a similar severity of illness in terms of their SOFA score, their acute physiologic score, and they had a similar mortality compared to other patients with pneumonia within our intensive care unit. What was different and what was really markedly different between patients with SARS-CoV-2 pneumonia and other kinds of pneumonia was their length of stay. So our patients with COVID-19 spent almost 14 days in the intensive care unit, whereas our median length of stay for other patients with pneumonia was 4 to 5 days. And this prolonged length of stay explains why this pandemic was so devastating to our healthcare system. Admitting 2 patients with SARS-CoV-2 pneumonia to your intensive care unit used up 1 bed for 1 month. And so this was really, it's just something that we were not prepared to deal with. So, the answer to your question, I think most people would have answered my question at the beginning, what's different about SARS-CoV-2 pneumonia, by saying that there is a cytokine storm. But this is actually sort of an urban legend that we watched evolve in real time. We actually had these data available to us very early in the pandemic, where these investigators compared the levels of inflammatory cytokines in patients with COVID-19 with other patients with critical illness. And they showed that there was actually no difference in those patients with COVID-19. I'm not showing you the data, but we found the exact same thing in our cohort, whereas all pneumonia patients had similar levels of inflammatory cytokines. But what was different that we found in patients with SARS-CoV-2 was related to this long length of stay. That high level of inflammatory cytokines in the plasma and in the BAL fluid was maintained over the course of the SARS-CoV-2 pneumonia. And so 14 days compared to 4 days. So you had what we think of as a cytokine monsoon as opposed to a cytokine storm. And we think that this prolonged exposure to inflammatory cytokines might explain some of the organ dysfunction and perhaps some of the long-term sequelae we've seen in the COVID-19 patient infected, in the SARS-CoV-2 infected patients. The other thing we found from looking carefully at the clinical data was that these patients had high rates of secondary bacterial infections, and this dates back to studies that were done by Anthony Fauci, where he suggested that many patients in the 1918 flu pandemic had died from secondary bacterial infections. We actually found in our cohort that about 45% of patients developed at least one ventilator-associated pneumonia, secondary pneumonia, and that these were associated with an increased risk of mortality. We think that this is probably just related to this long length of stay. So I'm going to just now turn to the molecular data, and although we have other data, I'm going to actually just highlight the single-cell RNA sequencing data and show how it really informed that model of molecular pathology that model of SARS-CoV-2 pathobiology that you saw on the initial slide. So here we're again looking at single-cell data. Again, we're not looking at very many patients. This is 10 patients with SARS-CoV-2 pneumonia with BAL samples that were obtained within 48 hours of intubation. On the left, we have the UMAPs that I hope you're getting used to looking at. Over here, we have our T cells, right? These are our T cell populations. On the right, we have our monocytes and macrophages. On the bottom, we have our B cell populations and plasma cells. And on the top, we have the small number of epithelial cells that we retrieve with this procedure. Just drawing your attention to the monocytes and macrophages over here. Those early recruited monocytes-derived macrophages are MoAM1 and MoAM2. These are those classical monocytes just coming into the alveolar space, and our more mature tissue-resident macrophages are sort of this hook on the tornado, these tissue-resident alveolar macrophages. So keeping that shape in mind, I want you to turn over here to where SARS-CoV-2 genes are found in panel D here. And what you're going to see is that reassuringly, they're in these infected AT2 cells, so we can detect the RNA virus with this RNA sequencing procedure. And they're also in those tissue-resident macrophages that I told you were infected, and in these monocyte-derived alveolar macrophages shortly after their recruitment. Not only these cells infected with SARS-CoV-2, but they're doing different things. So they're making more of these T-cell chemokines like CXCL10 and CCL4. And they are responding differently to the interferons that are present in the alveolar space. And you can see this sort of darker color in these infected cell populations, even though everything's dark because it's all responding to interferons. We've labeled this an interferon gamma response score because at the time that we're doing this BAL procedure, we detected no type 1 interferons in the alveolar space. That's not because type 1 interferons aren't there, but it's because we are sampling these patients at the time they develop respiratory failure, which is typically 5 to 9 days after their onset of symptoms. And where is the interferon gamma coming from? Well, it's coming from where you would expect it to come from these activated T cells. And so just pointing out that this small number of samples looking at this pathology at single cell resolution really gave us some insights into those mechanisms. So how does this relate back to our long length of stay? Well, this concept comes directly from work that John Butler did long ago when at the University of Washington. In this study that was published in JAMA in 1986, Dr. Butler challenged the widely held view at that time that ARDS was a completely homogeneous disease, and this was based on the radiographic appearance of the chest X-ray. So this group of investigators with Dr. Butler performed chest CTs on patients with ARDS, and he actually showed that ARDS is quite a heterogeneous and patchy disorder. And this has really shaped our thinking about the way that we manage these patients in terms of mechanical ventilation, and has shaped some therapies, motivating Dr. Gattioni to perform studies of prone positioning, which we routinely use now in our intensive care unit, taking advantage of this regional heterogeneity. When we wondered whether this sparse expression of ACE2 in the alveolar space, this less than 2% of cells being infected, might actually cause a spatially restricted infection that slowly spreads over the lung, as opposed to a more synchronous infection across much of the lung regions. And we found that was the case. This is an example of a patient that died shortly after presenting to the hospital. This patient decided to forego life-sustaining therapy And here you can see using RNAscope, we're looking at SARS-CoV-2, positive and negative strand DNA and CD206 to mark macrophages. You can see this macrophage that's infected here, an alveolar epithelial cell that's infected. And yet over in this alveolar space right next to it, you see no evidence of this SARS-CoV-2 infection. And so the hypothesis that we put forward is that these inflammatory circuits between these monocyte-derived alveolar macrophages and activated T cells happen within local lung regions, and that these infected monocyte-derived macrophages, which Paul Cubes showed are capable of moving through several alveoli through the pores of cone, act as a Trojan horse that helps to spread the virus slowly across the lung. And as a result, you get this sort of creeping forest fire kind of progression that causes this long length of stay, as opposed to sort of a conflagration that you have with flu. And that's just represented here on this top panel as a with the alpha-2,6 sialic acid receptors that are the receptors for influenza virus. They're ubiquitously expressed throughout the lung epithelium and the alveolar space. This causes a relatively short duration between the onset of symptoms and respiratory failure. Anybody that's an intensivist in this audience has certainly admitted somebody on some low level of oxygen with influenza and has had them with florid ARDS 12 hours later. And this relatively synchronous progression and recovery that we see in influenza with this 4-day course, as opposed to SARS-CoV-2, where we see this 5 to 9 day course between the onset of symptoms and the development of respiratory failure, then this long course of illness, followed by a slow recovery. And in patients that fail to repair their lung, we think that they go on to develop fibrosis, and we'll talk about that in just a moment. So we were really interested to see if this, this model is great, nice to talk about the pathobiology, but actually, can we translate that into therapies? Can this be actually used with any clinical impact? And during the COVID-19 pandemic, for better or for worse, clinical trial centers like ours, and I'm sure like yours, were inundated with the opportunity to participate in different clinical trials that were ongoing for patients with SARS-CoV-2 pneumonia. So we were selecting between a number of different trials in which to consider enrolling our patients, and we wondered whether we could use this model of SARS-CoV-2 pneumonia to predict therapies that might be effective. And furthermore, could we maybe use some of the technologies that we developed to actually do pre and post sampling in this molecular profiling to see if those therapies worked? The example I'm going to give you, or the drug that I'm going to give you that we really focused on, is a crack channel inhibitor. Crack channels are store-operated calcium channels. These are really important for T cell activation, and this was known from work that our collaborator Murali Prakriya, who discovered these channels, had published some years ago. And I had done some work with my colleague Gökhan Mutlu, that had showed that these might be important for macrophages. So we actually, thinking about our model, decided to participate in a randomized double-blind placebo-controlled trial of a crack channel inhibitor, Oxora, for the treatment of patients with severe COVID-19 pneumonia. And what's important here is that we had initially said no to this trial. We said we will pass because we thought it was a bad idea to be inhibiting T cells in a viral pneumonia. But after we saw these circuits in the single cell data, we thought that this actually might be a way for us to target these aberrant inflammatory responses. And we were able to partner with the company that does this work, Calcimedica, to actually do a pre and post study. So in patient, we did BAL before and after administration of the drug, and subjected it to those same procedures that I told you about that are available in the SCRIPT study. And I'm not going to show you all the data here. This is not yet published, but is getting close to publication. And this is actually just looking at the placebo group on the left, the drug group on the right. And here we're looking at the change in inflammatory cell gene expression between the beginning and the end of the infusion in the placebo patients. And you can see this increase in inflammatory gene expression in both the monocyte-derived macrophages and the activated T cells that was nearly completely abrogated in the drug-treated patients. So these data were really, it was nice to see confirmation of these findings in the larger multi-center placebo-controlled trial, phase 2 trial, where the company reported a 56% reduction in 30-day all-cause mortality in hospitalized patients with COVID-19. All of these patients had received corticosteroids, and many of them had received IL-6 receptor antagonists or JAK inhibitors. So this was a promising phase 2 result. So I'm going to start. So I'm going to go through and talk a little bit now about how this molecular pathology really helped to inform a clinical decision, not a therapy so much as not a drug therapy, but actually a surgical therapy for patients with COVID-19. And to talk about this, I'm going to introduce this idea of another cell population that was discovered by single-cell RNA sequencing, and that is these transitional cells. So these alveolar type 2 cells that I showed you in our cartoons before are also a progenitor cell population that divides after a type 1 cell is lost to reform this type 2 cell population and then differentiate into a new type 1 cell. And as that differentiation process occurs, there's a population of cells that accumulates that seems to be a bottleneck in this differentiation process that's been called many different names, but I'm just going to call it a transitional cell state here. And these transitional cells are never seen or hardly ever seen in the normal human lung. They are only observed in patients with pulmonary fibrosis. They've only been observed in patients with pulmonary fibrosis lung explants. And we think that these cells are actually this failed epithelial repair might actually be driving this fibrosis. And this is a concept that I think is being increasingly accepted by the field. So on the left here, you have this cartoon that's showing you these type 2 cells and type 1 cells and alveolar macrophages. This is just the same cartoon I showed you flipped on its side. In response to injury to these cells, there's this recruitment of these monocyte-derived macrophages that I told you somehow drive the fibrotic process. We're not sure exactly how. In response, these type 2 cells try to re-epithelialize the alveolar space, in the process forming these transitional cells. And if this process gets stuck here, these cells continue to get recruited and this inflammatory process continues. If these cells successfully heal, these monocyte-derived macrophages will undergo apoptosis, fibroblasts will return to quiescence, and the lung will heal. So the two things that were actually concerning that were preventing us from doing lung transplants in patients with COVID-19 was, first, there was a concern that there would be a recurrence of SARS-CoV-2 in the allograft. We weren't very worried about this because we had done hundreds of BALs at this point in time in the pandemic, and we were pretty sure that the SARS-CoV-2 was clearing at the time points we were looking at. These were patients that were on ECMO typically for 6 weeks or more. But the bigger concern was, are these patients actually healing, or do they really have something that looks like an irreversible lung fibrosis? So our surgeons started a warm autopsy program with us where patients who died from SARS-CoV-2, we took biopsies of the lung and we subjected them to single-cell RNA sequencing. Here you're seeing 2 of those warm autopsy specimens along with one patient, the first patient that underwent transplant. And Nick Markov and Melissa Querrey used a procedure called transfer learning to take those single-cell data and to map them onto that data set from Vanderbilt that I talked about, of patients with end-stage pulmonary fibrosis who met traditional indications for transplant. And what they found was that these cells mapped perfectly onto the pulmonary fibrosis cells, including the formation of those transitional cells, which I told you are only seen in pulmonary fibrosis, those monocyte-derived alveolar macrophages that we've been talking about, and a specific cell population of fibroblasts that is only seen in pulmonary fibrosis. And Melissa was able to confirm that in actually for the first 20 patients we transplanted using immunohistochemistry to look for those transitional cells marked by keratin 17 adjacent to fibroblasts. And it was really those 2 findings that informed our decision to perform the first transplant for patients with COVID-19 in the United States. It had been done in China and Vienna before us. And we subsequently did 45 of those transplants. Those patients have done well with outcomes. We now have 3-year outcomes on those patients, and they've all done as well as the rest of the cohort. Finally, I'm just going to conclude with talking about how these molecular pathology techniques can inform some of our mechanistic studies in mouse models to identify new targets for therapy and potentially to lead to biomarkers that might indicate who might benefit from those therapies. So this story comes from this story started with Satoshi Watanabe, who was a postdoctoral fellow in our lab and has now got his own laboratory in Japan. And what Satoshi did was he looked at single-cell RNA sequencing that Herbert Schiller in Munich had generated from the bleomycin model of fibrosis, where he did single-cell RNA sequencing essentially every 2 days during the bleomycin-induced injury. And what Satoshi noticed was that there was activation of a stress pathway, the integrated stress response that happened in those alveolar type 2 cells as they were differentiating into type 1 cells. And Satoshi wondered whether that was pathologic, so he used a compound that was generated by Peter Walter's group at UCSF called ISRIB to inhibit the ISR, and he showed that prevented bleomycin-induced or attenuated bleomycin-induced fibrosis in both young and old mice. And Satoshi went on to use a lineage tracing system where he marked the type 2 cells, that progenitor cell population, with a green label before he gave them bleomycin with or without this tool compound. And what he was able to show was that the number of lineage tag type 1 cells increased, so that differentiation process increased, and the number of those transitional cells decreased, suggesting that the way ISRIB was working was by inhibiting that pathway and accelerating that differentiation of the alveolar type 2 cell into the alveolar type 1 cell. And Sung-Hae Han and Navdeep Chandel took that observation back to our aging question that I started the talk with. And they were studying age. Sung-Hae, who is now, was at the time a K08 and Parker B. Francis Fellow, now has her own R01. And Nav were asking the question whether mitochondrial dysfunction that happens with age causes a defect in the regenerative potential of these type 2 cells. And what they found was that mitochondrial dysfunction leads to an increase in the NAD or NAD over NADH ratio, and that causes activation of this ISR that becomes pathologic with aging. And the hypothesis is that the reason that Satoshi's small molecule worked was that it was inhibiting this pathologic activation of the ISR. And so this suggests to us a potential therapeutic that might be used to treat patients who are developing this lung fibrosis after injury, or perhaps patients with some forms of pulmonary fibrosis that are accumulating these transitional cell populations. And we're then able to ask, can we think about biomarkers that might indicate patients that are failing the epithelial repair and might benefit from those kinds of therapies? And the thing that came to mind was these monocyte-derived alveolar macrophages. So remembering back here to the model, our hypothesis is that if these alveolar type 2 cells fail to differentiate into type 1 cells, we're going to get ongoing recruitment of these monocyte-derived macrophages. And those should be able to be sampled using a BAL procedure, and we should see that those are associated with this failed epithelial repair and the development of fibrosis. And the COVID-19 pandemic again gave us the chance to do that, this time with patients with long COVID who were presenting to our clinic, weeks or months after, actually it was all months after their initial COVID-19 injury. And this is a special group of patients with long COVID. These are patients who had persistent respiratory symptoms and had abnormalities that were detectable on CT scan. And when we used a machine learning approach in collaboration with George Washko at Brigham and Women's to look at these CTs in an unbiased way, we show that many of the abnormalities were fibrotic abnormalities in these patients. And in a subset of them, we were able to do single-cell RNA sequencing of BAL fluid. And again, you've just got the same sort of cell populations that you're familiar with now that you saw in the previous BAL fluid samples. And what we saw was that the abundance of these monocyte-derived alveolar macrophages, those pathogenic macrophages that we saw, was increased in patients with this persistent respiratory symptoms and radiographic abnormalities. And this actually nicely correlated with the fibrotic fraction of the lung, as did the levels of MCP1 measured in the BAL fluid. This is CCL2, which is the main chemokine that attracts monocytes into the alveolar space in response to injury. and highlighting that not only are these potentially a biomarker, but maybe also a target for therapy. We actually found that these monocyte-derived alveolar macrophages changed only in their abundance with the severity of fibrosis. So those patients that were getting better and those patients that were getting worse had essentially the same transcriptome within those monocyte-derived macrophages, suggesting that, going back to the first example, if we were to target these cells with the CSF1 receptor, that might also be beneficial in accelerating fibrosis resolution in these patients. So this is I hope that I've sort of convinced you of a potential model for how we can pursue this kind of science moving forward. All of us in all of our academic medical centers have high volume, high quality clinical programs that are generating a wealth of data in the electronic health record about disease phenotypes and disease courses. We are oftentimes, as part of the clinical care that we provide generating samples that can be subject to these molecular pathology techniques, be it single-cell RNA sequencing, transcriptomics, et cetera. And very often, instead of looking at those samples, we're actually throwing them away or letting them sit on the shelf. But it's now possible with emerging tools that are coming out in AI and computational sciences to start to think about integrating these data together into these types of models of disease, this molecular pathology of disease, and identify from those models potential targets for therapy and potential biomarkers for who might benefit from those therapies. We have the laboratories available to us to causally validate those potential targets in mouse models, mouse and cell models or organoids. And then by performing clinical trials of these therapies within our medical centers, where we're able to actually regenerate these multi-omic technologies, whether the trial is successful or failure, it will iteratively improve the next round of predictions. So I'm going to finish there and hopefully leave a little bit of time for questions. I again want to thank you for the opportunity to present this lecture. I want to thank all of the people that did this work. I've tried to acknowledge them as I've gone through the talk, but I just want to point out that these are all the authors on the COVID paper that I showed you in the middle of the talk. This really does take a lot of people working together to work this kind of science. And I want to thank our funding agencies and, of course, the patients and families who have generously consented to participate in these studies. Thanks, and happy to take questions.
Thank you so much, Dr. Budinger. There's a question from Dr. Werfel. So there were a significant number of COVID-19 patients that had early evidence of alveolar destruction and fibrosis, but then went on to repair this pathology, some completely. Have you learned anything that is unique about success in repair?
Yeah, that's a great question. And, it's a great question, and we talk about this all the time. We also have those patients in our cohort. We have patients on ECMO that look just as bad as some of our patients with transplant that went on to completely recover. The problem, and when we were looking at those patients, at least when they were very ill, at least at the level of BAL fluid, and the few examples we haven't seen differences there. We haven't published those results. That said, we tend to really undersample all the patients that are getting better. So, usually if you're getting another sample, if you're getting a sequential sample, it's because you're doing bad, right? And if you're getting better, we generally are not willing to put you through the risk of doing a bronchoscopy. So we really need to understand how the successful repair is happening in this minority of patients, because I think we can learn a ton there, but we just don't know. It's a great question.
And another question having to do with any host or substrate features that provided any predictive information about patients who were going to go on to have the increased concentrations of those monocyte-derived macrophages that appear to be driving some of the fibrotic lung changes that you're seeing?
Yeah, so we actually, Luisa Morales-Nabrada, who I actually mentioned in the talk, published a paper just last year with Ben Singer in Nature Immunology, where she was asking those questions specifically in the looking at markers of outcome in the BAL fluid, and it's actually in the T cells. So it was actually in the persistence of activated T cells, particularly CD8 T cells. And some of those T cells were actually, those T cells were preferentially reactive against open reading frames in the SARS-CoV-2 genome. So it seems to be, the simple distillation of that large amount of data is that T cells that are activated against inappropriate epitopes seem to be driving some of these circuits. And we really think that didn't show the data, but we actually think that the primary cell that's being targeted by Oxora is probably the T cell. So we actually think that that cell population is likely to give us more information than the monocyte-derived macrophages, which, as far as we can tell, are just sort of dumb soldiers that are doing what they're told.
Thank you. A couple other questions. So the clinical trial results of the crack therapeutic are very interesting. What are the next steps for this therapeutic, and what population would you study now?
So I can tell you what we are studying. I'm not sure it's what I would have picked, but this is not, we don't get to pick which trial. We're actually looking at acute kidney injury in patients with ARDS is what the company has decided to pursue. So we are doing a clinical trial. They're enrolling in a clinical trial looking at acute kidney injury in patients with severe pneumonia requiring mechanical ventilation. That's the next place they're taking this.
And from Dr. Morell, our interventional trials in pulmonary critical care have not incorporated molecular pathology, as you discussed, into their design yet. What do you think are some of the very first relatively easy steps trials should take in our field?
Yeah, I mean, I think we have to get, yes, and I actually see it as a problem in our field because we're actually going, I think, in many cases the wrong way. Where we have these syndromic definitions like ARDS that we actually make broader, right, with good intentions, right? But at the same time, by making those broader, we actually get away from exactly what we're talking about here. And then we have, other syndromic, even when we get to a definition like pneumonia, we use another syndromic definition, for example, community-acquired or ventilator-associated pneumonia, as opposed to thinking about sort of a pathogen-specific diagnosis. And I think, my personal call would be for, smaller trials that are focused on specific pathogens. I think that's the lesson we should learn from the pandemic. When we're able to actually get together a large number of patients with that are infected by the same pathogen, we're able to identify several different therapies that are effective in a pathogen-specific manner. So I think we should be going smaller and more granular as opposed to bigger and broader. But you'll find others in the field that will disagree with me.
And then from Dr. Garib, have you started to apply multiomics in your models to expand beyond the transcriptomics focus?
Yes. Well, yes, we are. We certainly aren't advocating that we would be thinking about single-cell transcriptomics as a tool for stratifying patients. What we're trying to do is actually use some of the markers that we're getting from the single-cell data and incorporate them into flow cytometry panels, which could be actually clinically actionable, or thinking about either cytokine or PCR-based panels that we could be looking to stratify patients into different post-response categories. We're not there yet. And in terms of other multi-omic technologies, our biggest push is in the spatial transcriptomics. Problem is that requires tissue, right? So we have to, but we think, so that's something that we're applying to autopsy tissues. The other place that we're looking at is actually in terms of metabolomics. I'm sorry, I should mention shotgun metagenomics for the DNA microbiome, actually seeing if we can actually get pathogen identification on the many patients in our cohort who clinically have pneumonia, are adjudicated as having pneumonia, but we don't have a pathogen identified. And then the use of both metabolomics and spatial metabolomics to see if we have effects from the microbe on the local metabolic milieu that might alter inflammatory cell phenotypes.
I'll give it another minute to see if there's any other questions from the audience. But thank you so much for a fascinating talk. That was wonderful.
Great. Thank you. Really appreciate the opportunity. Thank you, and have a great day.
And Scott, I'd like to thank you for such a nice example of a working translational science and team science that you brought to show the group here, and sharing it with us, and everything you do for career development and mentoring of young physician scientists.
Thank you. It's important. That's
It is.
Yep, absolutely. All right. Take care now.
Thank you,
Bye-bye.
Scott.
Thank you. Thanks, everyone.